Okay, so this is my introduction to the plant laboratory, and we're going to start with animals. Now, there's nothing that speaks out animal more loudly than Hox genes, and what I'm hoping is that you'll remember back to the module of instruction that you've just been through to just recall what Hox genes are and what they do. Okay, well, what are they? They're developmental genes. They're homeotic genes that determine the identity of particular body parts. And in particular, when you hear the term Hox gene, you should make the association between that term, Hox, and the anterior to posterior, that's front to rear, positioning of things you find along the body axis, the main body axis of an animal. In other words, this is what determines what kinds of things go in the front part of the animal versus the middle versus the back. And in particular, the idea is that in the body plan of your typical animal, we've got repeated parts, and so a little mistake there. And so, for example, in the body plan of an insect, we're going to have several segments that are going to be organized um, into different body parts, and so the animal might start off as basically initially what we have is a bunch of more or less identical segments. And in the front part of the animal, this is going to be the front, this is going to be the anterior, and this is going to be the posterior. We're going to see different body parts developing according to different rules that are set up in the different parts of the animal. So the classic example here is that if we're talking about a fruit fly, in the very front part of the body plan, if the segment gives the instructions to build an appendage, it's some type of structure that sticks out from the body segment. What's going to happen is that the segment will start building an appendage, and the appendage will develop in the form of an antenna. The antenna is basically the appendage that forms from the very frontmost segment of the body. In the next segment back, the segment gives a signal to build an appendage, and the appendage for the second segment is different. It gets converted into a mouth part, the parts of the insect that chews the food and swallows. Okay, and if you go a few segments back, and maybe the fifth segment back here, the segment gives a signal to build an appendage, and what you end up with is a regular walking leg. Okay, so we'll have three segments here. They're going to be producing walking legs on the insect. And this is effectively how an insect's body plan gets made. We've got different structures in different parts of the insect. Here we've got antennae, mouth parts, and walking legs that all start off with the same instruction, the same basic instruction: build me an appendage. And if we happen to be in the front part of the body, that appendage gets built as an antenna. Basically, there are a set of rules that are set up by transcription factors specific to the front end of the animal that cause the appendage to get built in this way, which is different from the way that the same instructions would be followed in the next segment back, or a few segments back. Those same instructions result in the full-on development of a leg. Okay, so this has all been very instructive in teaching us how development works and how development has led to the differences we have between different evolutionary lineages. Okay, so that's the animal part of the introduction. And what I want you to think about here are a couple of things. First, the idea that we've got anterior end-to-end positioning, anterior to posterior positioning, that are determined by specific Hox genes. The first Hox gene in fruit flies is called labial, and labial is basically the trigger that dictates the rules that are set up such that when we start building an appendage in the frontmost segment of the body of an insect, it turns into an antenna. Proboscipedia is the set of signals, is the trigger. It's actually the trigger that sets up the rules in the second segment of the animal that results in that same trigger of building an appendage to lead to the development of mouth parts. And so we've got a whole bunch of different sets of rules that are set up along the front to back axis of the animal. Okay. The other thing is the idea that yeah, there are rules, and these rules are basically determining what kind of appendage gets built. We're going to see both of these themes developed as we get into how this all parallels with what goes on with the development of a plant. Okay. And just as a little aside here, in the lecture, Dr. Bailey talked about Antennapedia, which was a mutant form of fruit fly that had legs growing out of the head instead of the antennae. What I have drawn here is a different kind of mutation. This one is maybe even weirder. Remember I said that the mouth parts are actually walking legs or appendages that are developed as mouth parts. That might seem a little bit weird to you, but how do we know that? It turns out that the reason why we're so sure that mouth parts are basically modified appendages is that in this particular mutation, if you do something to change the environment of that second segment there such that we don't get the proper signals to cause the appendage to develop into mouth parts, what happens is that we get those same appendages developing along what amounts to more of a default set of instructions, which is that they grow into walking legs. So yeah, this weird mutant fruit fly is found. It's got walking legs developing from where the mouth part should be, and this teaches us a whole lot about the way that animals are put together and how the different parts of the animals are basically modified from structures that you find in other body parts. Okay, well, that was a long introduction on animals, but animals are really a good place for us to start because we're going to be seeing something quite different and quite similar in the case of plants. And in particular, I want you to look at a flower. I want you to think about a flower really carefully and how on a flower, similar to a body, the body of an animal, we've got an axis of polarity. For animals, we have the anterior, the nose of the animal, and a posterior on the other end. In the case of a plant, what we've got is a base of the flower to the very tip of the flower. Okay, and there's along this axis of polarity, we've got different structures developing. Okay, so if we sort of visualize they're kind of being a main axis to the flower, closest to the base, we've got these structures that we'll call sepals. And the sepals are the things that you know about because if you ever have had a rose bud before it opens, you know that it's kind of green and closed like this. It attaches to the stem, and after it opens up, those green parts are kind of at the very base. Those are the sepals, and the sepals are at the bottom portion of the flower. And outside of the sepals, you've got these big feathery, showy, gaudy things, and those are actually attaching to that main axis further out towards the tip, not quite as basally as the sepals. And these are the petals. And the petals and the sepals together create the structure called the perianth, which are kind of the attractive portions of the flower. They're not really involved in reproduction. They're just kind of there, largely, I guess you could say the function of the perianth is to attract pollinators. The whole idea of a plant producing a flower at all is that it's successful. It makes a plant successful in having itself pollinated and its pollen delivered to other flowers. The reproductive parts of the flower are attaching further out towards the tip. And the next things you find along the axis of the flower are the stamens, which are these filaments. And these filaments will have little anthers at the end. And these are the structures that will carry the pollen. Okay. Now at the tip of the flower, what we have is the pistil or the ovarium or the gynoecium. We normally draw the pistil kind of like this, where you've got an ovary at the bottom, a style, and then a stigma, the sticky part, at the very tip. But this ovarium or pistil is really made up of several carpels, which are modified leaves that are fused. So this is a carpel over here. This might be a carpel over here. This might be a carpel over here. And if we put them together, if we have them close in, what you end up with is this kind of vase-shaped structure that is entirely closed. And so even the pistil of the flower is actually made up of several modified leaves, which are the carpels that are fused together to create the pistil. And so along this axis, going from the base to the tip, we actually have four separate sets of leaves. I'm going to use the term verticil, okay, to refer to these different populations of leaves, the different sets of leaves. So the verticils along a flower would be the sepals at the bottom forming the calyx. You've got another verticil, which are the petals forming the corolla. You've got the next verticil out, which will be the first reproductive verticil. These are going to be the ones that have male function. These are the stamens and the carpels, the structures that we have forming the ovules. I haven't written that term, carpel. I think it's spelled with an E. Carpel is the part in your hand that you can get tunnel syndrome on. But these four sets of verticils are in a linear array along the axis of a flower. Okay. Now, a little backing up a little ways, we're going to be thinking about different kinds of stems because it might be a little bit difficult for you to think about this, but I'm going to tell you that a flower is basically a modified vegetative stem, kind of like this guy over here. You've got the main stem. This is where the plant's growing. You've got the leaves attaching to the stem. These structures here are called petioles. You've got leaves out here. And the spaces in between where the leaves attach, this is called a node. And the nodes are where the leaves attach. The space in between are called the internodes. And so the anatomy of a regular vegetative branch, or a branch that has regular photosynthetic leaves, is I don't think it's going to be terribly shocking or new to you. This is the way that plants grow. Okay. Now, for students, it actually is usually a little bit easier for people to picture the idea that a pine cone, what I have drawn over here, it's our photographed over here, a pine cone is really a modified branch. So these structures over on the left and over on the right are really based on the same theme. We've got a central axis. We've got a central axis, this is kind of like the main stem, and we've got leaves attaching. Only in the case of a pine cone, the leaves that are attaching are modified in a way that's related to the plant's reproduction. And you might even know this. If you've ever taken apart a pine cone, you pull out one of these little bracts. At the very base of the bract, you've got a couple little pine seeds. And that's that basically is kind of telling us that what we have here is a specialized branch. It's a branch that's specialized for reproduction, and in particular the leaves, the modified leaves on here, in botany class we call them megasporophylls, but these modified leaves are related to female reproduction. Okay. These are not the parts of the plant that produce the pollen. The pollen is produced elsewhere. In the case of a pine tree, the pollen producing structure is also a cone. These guys are cones as well. We would call these male cones, and if you were to take one of these male cones, split them apart, you would find that, yeah, there's a central axis, and on the central axis we would have modified leaves. In this case, the leaves are much smaller, but each leaf is going to be producing a whole bunch of pollen grains. And so if you go out and look at a pine tree in the springtime, what you'll see are these really tiny, almost look like corn on the cob like structures, but early in the spring, if you were to shake a branch, you would see a whole bunch of pine pollen drifting away from this. And that's because we've got a whole bunch of specialized leaves on this modified stem, each one of which is producing the pollen, as opposed to producing the seed-like parts. Okay? So yeah, I think that when I tell your typical student that a flower like this is really the same as a structure that we have over here, I get a lot of strange looks. But if I tell students that a pine cone is simply a modified branch in which we don't have regular photosynthetic leaves, but we have leaves that are specialized for reproduction, that's generally a whole lot more believable. Okay, and so let's start there. But let's ask the question: well, how does a branch know to become either vegetative or reproductive. So for example, if you've got a tree like that's growing up like this, and you might have some branches that are going to be produced, and some of these branches are going to have regular leaves, and these are the ones that are going to have photosynthesis, all well and good. And we need to have lots of these vegetative branches, okay? And so how do we end up getting a branch over here, right here, that's going to be different? It's going to be producing a pine cone, maybe a male cone or a female cone. Well, I'm glad you asked me that because we know. It turns out that in the same way that an appendage can grow into an antenna in some parts of the insect, a mouth parts, the mandibles in another part of an insect, or the legs in a different part of the insect altogether, you could have a bud of tissue develop either as a vegetative branch or as a reproductive branch, depending on whether or not we've got the appropriate rules set up. And in this particular case, there's a gene that's related to hawks. It's called leafy. And leafy is the one that is the determining factor to decide whether a little branch is going to develop as a vegetative unit or primarily a reproductive unit. And so if leafy is on, so basically the question goes, is leafy present? Yes. Okay. Then we develop as a reproductive structure. If leafy is absent, then we develop as a vegetative structure. So yeah, what determines whether a branch grows up like this and is vegetative, or grows up like this or like this and is reproductive, is determined by the presence or absence of transcription factors that are influencing the way that the plant develops. Okay. So yeah, that's part one. It's actually very similar to what goes on in the case of an insect or a mouse or any of the animals that are following the rules set up by the Hox genes. Okay. Now, you might at this point, some of you might be thinking, well, what's going to determine what? Let's say we do have yes here. Okay. So let's say we do have the decision that we're going to be developing as a reproductive branch. Then the question goes, are we going to become male? In other words, are we going to be producing staminate cones like these guys down here? Or is it going to become female, like the pine cones that will actually produce the seeds? Those are the ones that we normally call pine cones. And basically there must be a different set of rules that will determine, once we establish that we're going to be reproductive, there are different rules that will determine whether those cones are going to develop as female or male. Okay. Now, what I'm telling you is that the flower, the structure here, is basically developing along the same sets of the same operational rules. And in this particular case, we've got a series of genes that we know that are active to allow us to have a base to tip polarity on this reproductive branch. See, what we lack in the case of a cone, what we lack in the case of a vegetative branch, is any kind of differences along the main axis. Pretty much the leaves that we have at the base are the same as the leaves that we have in the middle, which are the same as the leaves we have at the tip. These reproductive leaves, the bracts on a pine cone, are pretty much the same if we're looking at the base towards the tip. We're not really seeing any changes as we go from one end to the other. Now in a flower, that's different. Okay, so I want you to sort of visualize, here is our axis. So here is our maybe an ancestral cone-like structure, and we have a zone at the bottom that is going to be the area where we want the sepals to develop. And then we've got another zone up here where is the zone where you want the petals to develop. And the next zone up is going to be the zone where you want the stamens to develop, and close to the tip is where you'll have the carpels develop, because that's the sequence that we want. We need to have sepals down here, petals in this area, stamens closer to the tip, and at the very tip we need to have carpels developing, because that's the part that will form the pistil or gynecium. Okay. Well, guess what? We know pretty much how this works. and I'm going to use a little bit of color coding to show you. All right. Okay, so we've got three different genes. We've got gene number 1, that's what will be called the A gene, A, which is present at the very base of the flower, but it overlaps the first two parts. So the A gene is active. It's actually it's expressed. We can find A gene activity in the bottom two parts of a flower, of a developing flower. The B gene, I'm going to draw it yellow, is active overlapping parts 2 and 3. So this is going to be the B gene. And the C gene is going to be active closer to the tip, going all the way to the tip. And so basically what we're generating here are three different areas of activity. In the bottom part of the flower, down this area here, we have A only. There's only A present, and under the influence of A only, the leaves, the verticils in that area develop as sepals. In other words, we give the signal, build me some leaves, and because we have A only present in that portion of the plant, those leaves develop as sepals. Okay? In area 2, we've got a combination of A and B, and with the combination of A together with B, that same signal, build me some leaves, results in the production of petals. Okay. Now, as you might be guessing already, in the area further out towards the tip, the area where we have the stamens developing, we no longer have A, but we've got B, and we're also picking up some C. And so we've got the influence of both the B and the C genes, and that's the signal that's required in order to develop the male portions of the plant, the stamens. And out towards the tippermost part of the flower, we have C only. And that's this is a signal that will develop as the carpels. Okay. We've all of this information is actually has been confirmed with laboratory studies using kind of little crest-like plants called Arabidopsis. Arabidopsis, which is a little cress. It's like a little mustard. And Arabidopsis is kind of like the fruit flies of the plant world. And what we found is that we can find modifications or mutations of the A gene, and we call those genes I think apetala. Actually, it's apetala too, because there are three versions of the apetala gene. And so each one of these genes, the A, B, and the C gene, have actually been identified, and their effects are known. It's not simply a model, it's something that actually is confirmed as being the mechanism of generating the different flower parts, at least for some kinds of plants like Arabidopsis and a few others. Okay, so think about this. When you undergo your dissection of a flower, where you'll have sepals and petals and stamens and some kind of gynoecium. Sometimes it's going to be underneath the gynoecium with the stigma over here. As we undergo our dissection of a flower, keep in mind, one, this is actually a vegetative stem that's been or a non-vegetative stem that's been modified to have a reproductive purpose, right? Second thing is that we've got this ABC determined polarity from the base to the tip. And in a lot of ways, this is basically a very nice supporting lesson to what you guys have learned in the lecture sequence in relationship to the Hox genes.